Well, welcome back. We've been learning about the language of life that's found in nucleic acids, specifically the DNA molecule and how that gets translated into RNA. And as you can see on the screen here, that's used in making protein. I want to go in this segment, as we did in the last, into some of the details of how these processes work. How does the information in our genes, in our chromosome, how does that get converted into traits that you and I can see? In our last segment, if you remember, we looked at something called the central dogma of molecular biology, and you can see that it includes three processes that we briefly outlined in our last session. Replication is when DNA gets copied once during the cell cycle, and so that every cell has a copy of all the genetic information that organism will have. And then throughout the lifetime, there are many instances in which some of this information is going to be copied into a molecule, another nucleic acid known as RNA, ribonucleic acid, that will be used in the production of proteins that you can see here. And so these processes have come to be known as replication in the nucleus, and then transcription also in the nucleus in the making of RNA, and then translation, which is the actual production of proteins that are out in the cytoplasm, in the ribosome specifically. And so this one flow of information from the nucleus, DNA, all the way out to the cytoplasm in the production of proteins is what has come to be known as the central dogma of molecular biology. Now, in this segment, we're going to focus on two concerns. Before we can look at translation, we really need to understand something about the language of life. And then before that as well, to understand how the different kinds of RNA, the roles that they play in the process of translation. Then finally, we're going to look at exactly how translation occurs. And then I'm going to leave you with a particular challenge for you to think about. So let's get started. What do we mean by the language of life? Well, it gets down to the concept of a codon. And this would be something worth writing down. A codon is really the words that are in the language of DNA. Just as you and I combine letters to form words in our native language, the native language of DNA has words as well, which we refer to as codons. But it's a language that it's made in the form, not of letters actually, but in nucleotides. And one such example of these nucleotides you can see in this very tiny segment of messenger RNA on the screen. UAG is going to provide the coding for a particular amino acid that is going to be used in the production of a protein. There are 20 amino acids that are used in the production of proteins. And so all these are going to be coded for through this process that we call translation. And so, in essence, that's the language of life. And when we see how these three-letter codons are translated into amino acids, we use something like this table to help us to decode and translate a nucleic acid in messenger RNA, those codons into the actual amino acids that form the protein. And like I said before, in this language of life, there are letters and there's also syntax. There are stop codons that act as a period in a sentence. And there's also start codons, like the one here that codes for methionine. All right, now with that brief introduction to the language of life and the way in which messenger RNA is used is that there is a taking of three nucleotides at a time in reading these codons, and we refer to this manner as a reading frame. The ribosomes are going to read this string of nucleotides on messenger RNA, taking them 3 at a time. And so if there's a shift in messenger RNA that changes only one nucleotide, you can see that that completely alters the amino acid that's coded for in that sequence. And so this is how messenger RNA is used to code for protein. But there are 2 other forms of RNA that play their roles as well. Let's look at those. And first we can see the form of RNA that we have before us is transfer RNA. At the very base of this, we have a complementary pattern of nucleotides that are going to bond to messenger RNA in the translation process. And because these complement the codons, we refer to these as anticodons. So in a sense, they are opposites. At the top of the transfer RNA molecule, we can see joined to this a specific amino acid for which this transfer RNA codes. And so because there's 20 different amino acids, there's going to be 20 different versions of tRNA. And then next, within the ribosome, we also have ribosomal RNA. A ribosome, very briefly, kind of acts like a zipper. There's a back to it, the small subunit, and then there's the top unit that has the binding sites that themselves are made up partly of ribosomal RNA. And so these are the binding sites that will perform the reading frame function that we mentioned a moment ago. And then the backside, or the small subunit, is what's going to hold the messenger RNA process in place during the translation process. Well, I'll leave you to think about that. And once again, thanks for watching. Bye-bye.